To kind of close our session, we have Antoine Ruzette from Harvard Medical School. He's going to tell us about how to build large synthetic data sets of microscopy images. Thanks a lot, Antoine.
Of course. Yeah, I think this is a perfect flow of information right now that we're really touching upon some sort of the biggest problems right now into training those big microscopy image data sets to train and fuel the next large vision models. And so my name is Antoine, and I'm a computational biologist with Sean Magison and Simon Norelica in the Department of Systems Biology at Harvard Medical School. And so for the next 10 minutes or so, I'd like to discuss about a framework we're working on to generate large synthetic data sets of microscopy images. And so what really got me thinking about this project in the first place is that need of large synthetic data sets in this age of AI. And so as we enter this age right now, one of the great challenge in computational imaging is to keep up with the data quantity required to train large vision models in a supervised manner. And as a matter of fact, vision models such as transformers learn from millions of object pairs. And so such models, though offer great promises in the future, they scale very efficiently, meaning that their performance will significantly improve with more data and more compute. They can capture rather long-range dependencies compared to the limited receptive fields that convolutional neural networks, classical deep learning models so far have showed. But the lack of inductive bias in transformers mean that they learn patterns somehow from scratch. And that requires much more data to actually generalize meaningfully. And so to give some numbers here, like the data set, the size of the data sets required to train a transformer range from 10 to the power of 6 to 10 to the power of 8 images. And in comparison, training a typical CNN, such as a UNet, will require 10 to the power of 3 to 10 to the power of 5 or so images. And so there's really a 10 to 1,000 here fold change required in the size of the training sets to actually enter this new field of transformer-based architectures. And so the question really that I ask myself is, how do we now get these ground truth data sets to fuel large vision models for microscopy images? Well, of course, the first thought that I got in my head is manual annotations. I don't want to do it personally. I don't want to spend careers and careers of people. That requires tremendous resources and money to actually annotate the quantity required manually. And so I think synthetic data here emerges as a rather elegant solution to try and tackle that along the way. And so to just backtrack here, I just want to ask this initial question of what actually makes a microscopy image different. And so what do we need to think about when we actually generate those images from first principles? And so, well, first it was obviously taken by a microscope. And so its formation and acquisition are based to some very specific physics of the system. And to only name a few here, the optical system, the physics of light of imaging, the illumination systems, meaning at which wavelength you're going to image your sample, the properties from the detector and the sensor. All of these systems will basically dictate, the final signal-to-noise ratio and overall quality of the image of your samples. And then, of course, microscopy images capture the morphology and organization of cells. And so that includes their shape, their location, their subcellular organization, the density of those cells in the tissue, and so many more cell properties that we also need to address in order to have something that tackles the realism of microscopy images. And so in this talk, I really want to address those two key drivers making up a final microscope images. And so basically here, my talk is going to be some sort of actually going back to first principle forward models and how that can help into making those data sets to actually go then into this age of AI. And so over the last year or so, we have developed tools to first simulate collections of cells and their biophysical properties so that we can address the realism of the sample. And second of all, we have developed a tool to simulate the microscopy image formation and acquisition systems to address the realism of the noise, of the artifacts, everything that makes up a microscopy image. All right, so we've developed Goo, which is a Python library to simulate cells in 3D. And it's fully open source. And I just want to introduce here some general concepts we've implemented in this model. And so a cell in Goo is a soft-shelled mesh that's pressurized with fluid. And the mesh itself can deform, and that deformation itself is being tightly controlled by its surface tension, controlled by its stiffness. Then cells can interact directly with one another through cell-cell adhesion, which can be homotypic or heterotypic. Cells can also move, typically following some random walk or persistent random walk. Then cells can grow and regulate their volumes based on a pressure-volume feedback loop. Cells can divide in two, either based on volume or time. And then some sort of going from this like physical models to more molecular cues, we also made this library so that cells can evolve in a discrete concentration grid. They can sense the metabolites in their pericellular environment. They can basically transduce that signal through a set of gene regulatory networks, and from which the output, the gene concentration, can then feed back and modify the cell functions over time. And so, we have implemented this rather comprehensive deformable cell model in modern user-friendly and Python-centric tools. And the most important one here being Blender, which is an award-winning computer graphics software. It's a beautiful piece of code, and it's been developed for the last 25 years. It's tremendous at handling a large amount of 3D meshes. And so, I just want to point here, obviously, that there are many ways of modeling biological systems. This is just one flavor of them, and that really, its usefulness is very dictated, obviously, by how you use it and the question you're trying to probe with it. And so here I'm just giving some example, the first one being a single cell dividing over and over until forming a compact clump of cells. That's about 256 cells. And so in the lab in general, we're very interested in studying zebrafish embryogenesis. And so we've also had simulations of that running on top of a yolk of a zebrafish and all of that as well. The second example, we then seed cells in a sphere, let them grow, divide, and move, and see and assess basically how much they mix, given variations of both their motion and the adhesion between those cells. Last example, it's I want to highlight here basically an example just taking a reproculator from the famous paper of Mike Elowitz, and how we linked basically the concentration of repressor 1 to the speed of motion. And so when cells are in blue here, they are in this like rather low speed motion, and when they're in red, they start jamming together, and like the speed of this motion increases. All right. So now that we can simulate developing tissue in 3D, the question is how can we actually tackle and simulate microscopes? And that's exactly what my colleague, Tali Lambert, at the Core for Imaging Technology and Education at Harvard has pioneered, and he's made a great library. And I mean, I here put a picture of him as if it was taken through a scope. And so, I mean, just imagine the head of Tali passing through a scope, and I'll just go through an overall description here on this library called MicroSim, which is a Python library to simulate realistic light microscopy images. And I will just go over the different sort of simulation stages that happen in the background of that library. So first, we have a ground truth stage that's responsible for generating the position and number of the fluorophore in that 3D space. And that's really where you can pretty much put anything you want. If you want to like write squiggly lines and pass them through a scope, you can. Of course, we want to do some cool things and actually have realistic shapes in there. Second stage is we calculate the rate of photon emission in collection for each combination of fluorophore, and so you can have as many channels as you want in here. And we also do the optical configuration in the simulation here. And this is all as a function of the wavelength that we sample our image at. Then the next stage, we actually scale the fluorophore distribution by this respective emission photon fluxes. And then this is being convolved with the optical point spread function of the microscope to form this optical image. And in the last stage, we go from an optical image to an actual digital image. And so really this stage like includes the addition of the noise due to the discrete nature of photons, some read noise due to the imprecision of detectors, as well as, of course, downsampling to the dimensions that we know and are aware for this like final image resolution. All right, so now the question that we've done is we can, of course, put our perfect masks here, or at least the fluorophore distribution that we get out of our simulations in the 3D space, in the 3D ground truth space in microsim, and then actually running the whole simulation. Here I'm illustrating this with a confocal setup with some parameters that you will, that you will recognize: the pinhole size, the exposure time in microseconds, in milliseconds. Sorry. So this is all parameters that we can really finely control now. And so, as a summary here, we have developed a suite of biophysical models to generate large data sets of highly realistic yet synthetic microscopy images. We have now tools to simulate physically plausible cellular morphologies and developing tissues with Go. We also have now a tool that allows us to tune basically all the knobs of a microscope in silico, simulated from first principles. And we can really put these two together to simulate the whole pipeline from as if you were taking a sample, passing through an in silico microscope, and then having this ground truth and the final microscopy image fully simulated. And the most beautiful part of that is that we now have generated we have generated basically millions of synthetic microscopy images, great diversity. We can pretty much simulate anything we want, of course, based on the constraint of some models. But we have, so the ground truth, we have the final microscopy images, but we also have all the optical parameters that with which the microscope were set up, right? And so, now in my opinion, that's really where now the fun begins, even though, it was already really fun, obviously, to do all of that in the first place. But so the big test right now that obviously I wish I was giving as a headline right now is to actually train a large vision models on this really rich data set to actually check whether it improves our algorithms or not. Like we were talking before about, we actually need those like 3D plus time massive data sets of microscopy images. Well, I want to argue that we have a first step towards that, and it would be so interesting to actually see and understand whether our algorithms can learn now across 3D and time with that sort of data set. The last, another piece that I'm really interested myself in is to have this like physics to image generative AI. And so I really want to train those diffusion models to generate microscopy images on those physics-based models. Meaning that right now diffusion models appear a little bit as like a magic painter, but I really believe in the future with data sets like that we can actually constrain the diffusion process with physical parameters, so that it's not just painted based on those images that I've given you, but it's actually transfer the style of a microscope with that set of optical parameters. And then I really believe tools like that are going to be great in terms of evaluating the bunch of image analysis pipelines we have, and not just as a way of testing cell segmentation, quality, and all of that, but really testing limit cases, right? Where, for example, we could in the future have an idea whether CellPost, for example, performs well across the full range of microscope parameters, or across a full range of density of cells or size of cells. And so right now we basically have tools where we can do some sort of like this like 360 evaluation tool across any parameters you might think of. All right, and so to conclude here, these are both open source community developed libraries. If you want to get involved, absolutely, please create a PR, start an issue. Like this has been driven for the last year with many open source collaborators, and decided to highlight libraries in this. And then I really want to acknowledge, obviously, the people I work with on a daily basis, mostly Sean Megason, who's my PI, as well as Simon Nohalika, and of course, Tally, who is just across the corridor of my office, who I probably go to knock at this door a little too often. So thanks so much.
There's a lot of really fascinating stuff that